Students, Assalamu alaikum. Hope you will be fine. Today we are doing exercise of chapter number 10, respiration. The first question is, what do you understand by respiration? You already know respiration is the oxidation of food in the presence of oxygen to release energy. In B part, they are asking, describe how you would show that a green plant respires. So here on page number 168, an experiment is given which is in test yourself. Okay, so you will not write the full experiment, just you will write the part which is required. We need the portion that green plant respire. So how we can prove that green plant respire? So here they are showing that we can use an indicator. Indicator is a solution which changes its color in the presence of oxygen or carbon dioxide. Okay, if oxygen is present, it will become alkaline. If carbon dioxide present, then it will become acidic, and when it will be acidic, it will be yellow. Okay, so here we are going to prove that when green plant respires. So here just we will say that we have this indicator which is red in color. Okay, so here in this indicator green plant is given. Okay, so what will happen, we will keep this apparatus for some period of time. This green plant will use all the oxygen which is present in the environment for respiration. So it will release carbon dioxide. So when it will release carbon dioxide, so this medium will change to acidic and its color will change, and that will be yellow in color. So this proves that green plant respire to release energy. Okay, then the next part of this question is, here I will come back to the exercise. Okay, so here next question is question number 2. What is the importance of respiration to living organism? So here you will write that you already know its importance. You can write in detail that respiration is to release energy, and this energy is utilized in all activities, even in the blinking of our eyes, in the respiration, in breathing, and doing any activity in our body or performing any act. We need energy, and this energy is just available by respiration. So you will write all these things in detail. Okay, now B part is, how does aerobic respiration differ from anaerobic respiration? So you will make a column and you will write aerobic respiration on one side, anaerobic respiration on other side. So first of all, you will write the difference of their definition, that aerobic respiration take place in the presence of oxygen, and anaerobic respiration take place in the absence of oxygen. Secondly, you already know that in aerobic respiration large amount of energy releases, while in anaerobic respiration a very little amount of energy releases. The last difference is of their end product. End product of aerobic respiration is carbon dioxide and water along with oxygen. Carbon dioxide and water along with a large amount of energy, while end product of anaerobic respiration can be of two types. As you already know, anaerobic respiration is also called alcoholic fermentation, in which end product can be ethanol plus carbon dioxide plus small amount of energy. Secondly, anaerobic respiration, when it is taking place in the muscles, at that time lactic acid and small amount of energy is the end product. Okay, then C part is, describe an experiment to show that yeast can respire anaerobically. So here what you will do, here on page number 167 an experiment is given and you will explain this experiment. So here you can see we have a test tube in which we have water. First we will boil the water, then we will add the yeast for boiling. What does it is a purpose of boiling, that when water will boil, so all the gases which are diffused in the water will release. mean carbon dioxide, mean oxygen which is present here in this water, that will move out. Okay, then we will cover this water with thin layer of oil. This is not just a simple water, it is a glucose solution. Okay, so this one is a glucose solution with yeast. Yeast are unicellular fungi. Okay, so what happened, here we are having a delivery tube. This is here in a test tube which is having lime water. Why we are using lime water? Because lime water turns milky in the presence of carbon dioxide. Okay, now what will happen? As oxygen is not present, so yeast will respire and it will release carbon dioxide, so along with ethanol. So what will happen? This carbon dioxide will move here and it will turn this lime water milky. So that proves that yeast respire anaerobically. Okay, now in the next question, so come here the diagram is given, and in this diagram, question number C, you can see here the diagram of alveolus, and this is the blood capillary. Okay, now the question is, the figure above shows a diagram of an alveolus. So this one is an alveolus and the blood supply. This one is the blood supply. Now the question number 1, or A part of this question number C, is name the structure label A. You already know this one is alveolar wall or layer of epithelium. Okay, basically we are calling it as alveolar wall. It is not basically the actual wall, as it is the outermost boundary, so we are calling it wall. As you know, in animal cell, wall is absent, so it is an alveolar wall. Okay, now B. What is this B? Inside this alveolus is a thin film of water. This B is thin film of water. And C, what is this C? This C is red blood cell. Okay. Now they are asking, name the large blood vessel through which blood travel from heart to the lungs. From heart to lungs, blood travel through pulmonary arteries. Okay, or artery. Okay. C part, question number 1 is, name the process in which oxygen and carbon dioxide enter and leave the blood at an alveolus. You already know, due to the difference of concentration, here oxygen will move inside these capillaries and inside combined with red blood cells, and carbon dioxide will move out. So the process by which it is moving in and out is diffusion. Okay, for this process to work effectively, there must be difference in concentration between the gases in the blood and the alveolus. Give 2 ways in which these differences are maintained. Okay, what are the 2 ways in which these differences are maintained? These differences are also given here on page number 173. These 2 differences are a continuous flow of blood through the blood capillaries and breathing air in and out of the alveolus. As this air moves in and out, so every time a fresh air is coming with every breath, so due to that this difference of concentration maintained. Secondly, this blood is flowing into the capillaries, so when this blood is red blood cells, they are filled with oxygen, they will move forward direction and new blood will come. So these 2 are the gradient difference due to which this exchange is occurring. Okay, the next question is state 3 adaptation shown in the diagram which enable the alveolus to function efficiently. Okay, now 3 adaptation. So what are the 3 adaptations? You already know, here they have shown you the wall of alveolus, which is 1 cell thick, mean very thin. Secondly, a thin layer of moisture, it is the second adaptation, due to which oxygen diffuses quickly into the thin film of water, then it exchange through the move from alveolus to the blood capillary. Third, you can see here adaptation that blood capillaries wall is also one cell thick, mean very thin. So these three adaptations are shown here in the diagram, which you can explain. Then they are saying in which part of the blood and in what form are the two gases are carried to and from the lungs. Okay, you already know carbon dioxide is carried mainly in the plasma. So here you can see here into the plasma portion carbon dioxide diffuse and as hydrogen bicarbonate ion or hydrogen carbonate ion. You can say bicarbonate ion or hydrogen carbonate ion. So it is in the form it is mainly carried in plasma as hydrogen carbonate ion. Okay, oxygen is carried mostly in the red blood cell as oxyhemoglobin. Okay, now the next question is describe how oxygen from the atmosphere get into the mesophyll cells in the green plant. So here you will explain the structure of leaf, how it will move. So you already know as due to the difference of concentration in the air amount of oxygen is more, inside the leaf the amount of oxygen is less. So from outside this oxygen will move inside through the stomata, and here around the you already know around these mesophyll cell thin film of water is present. It will absorb into this film of water, then it will enter into these mesophyll cells, and then the process of respiration will take place. So basically due to this gradient difference it is moving in. Okay ji. Okay, now the next is that in what way the lungs adapted for efficient absorption of oxygen. Okay, in what way lungs are adapted? So already know lungs adaptation again in the alveolus. You already know here the structure is given, and which is again you will explain all these things. Thin wall of alveolus hai. Then thin film of moisture is also one of the second reason, and third one is thin richly supplied with blood capillaries. Lot of capillaries are present, so and they are also one cell thick, so quickly exchange take place. Then ventilated air which is coming inside and continuous flow of blood into the capillaries is the reason due to which lungs are adapted for the gaseous exchange. Okay. Next is in what form oxygen and carbon dioxide carried in the blood. Same as we have explained here, that carbon dioxide is mainly through plasma in the form of hydrogen carbonate ion. Oxygen is with the red blood cells and it is in the form of oxyhemoglobin. Okay. Now the next question is, how does expired air differ from atmospheric air? Account for these differences as far as. So you already know here a table is given in the book on the page number 175. So here you can write all the differences of oxygen, that in inspired air 21% oxygen and in expired 16.4%. Then the percentage of carbon dioxide in inspired air 0.03%, in expired air 4%. Nitrogen remains the same as our body is not taking it from the air. Water vapors, you already know their amount is variable in the outer air. In inspired air, air can be dry, air can be moist according to the environment, but expired air is always saturated. Temperature of the outer air or inspired air can be different. It can be according to the environment, but expired air temperature will always be according to the body temperature. Dust particle are, you already know, filtered by cilia in nasal cavity and in the respiratory tract. So in inspired air variable amount of dust particles can be present, but in expired air very little if any. Okay. Now the next question is that okay, again this is related to a diagram. So here a diagram is given. So they are saying this diagram shows the operators used to investigate the release of energy in a living organism. The P had been rinsed with an antibacterial agent. Okay, here P's are given in this operators, and they are cleaned with antibacterial, so no bacterial activity can take place. So if bacterial activity will occur, again the process of respiration will occur, and amount of energy can release. So to avoid all these things, it is cleaned by antibacterial agents. Okay, the question is, why is a vacuum flask used in this experiment? I already explained you that we are using vacuum flask. Vacuum flask, this is the glass portion of the vacuum flask. This one is the outermost plastic portion of the vacuum flask. Between these two, a vacuum is created, and this vacuum is to prevent the heat gain and heat loss by conduction, convection, and radiation. Okay, then the next one is, why is it placed in inverted position? So here it is in inverted position or upside down. The reason is that these P's should come in contact with this thermometer, so the increase in temperature can be indicated on this scale. Okay, then after the apparatus is set up, the temperature rises steadily by 3 to 4 centigrade each day. How does this measurement indicate that the pea seeds have released energy? Okay, so as they are saying that after setting this experiment, every day the temperature is rising 3 to 4 degrees centigrade. So this is showing that respiration is taking place, energy is released, and this released energy is in the form of heat, and this is increasing the temperature, which can be measured on the scale. Okay, now the last question is, the temperature of the pea seeds rises in the day and drop a little each night. How could you be sure that this is not simply related to the daily fluctuation of temperature, as the energy has in fact been released by the peas? Okay, you already know we are using vacuum flask, which is preventing the heat gain or heat loss from the outer environment by conduction, convection, and radiation. So it is proof that there is no such thing due to which from environment heat can gain or loss, and the only thing due to which this fluctuation can be due to experimental error. Okay, so do complete your exercise. If you have any problem, you can ask me, and I hope that you all will complete your work. Okay beta, wish you all the best. Allah Hafiz.